So I think we'll get started now. I want to remind everyone that after the lecture there will be a reception in the library sponsored by FAES. And I would like to introduce today's speaker, Dr. David Bartel, who visits us from the Whitehead Institute and the MIT in Boston. My name is Marcus Hafner, and I am an investigator at NIMS since 2 years. And one of the reasons I'm happy to introduce David is that before I came to NIH, I was a postdoc in Tom Tuschl's lab at Rockefeller, who, as it turns out, made the discoveries he's best known for pipetting in David's lab. So David Bartel graduated with a BA in biology from Goshen College in 1982, and then took off a few years to do voluntary work in Africa. And then continued his education as a graduate student at Harvard with Jack Szostak, where he obtained his PhD in 1993. With Jack Szostak, David used in vitro selection methods to select ribozymes, basically enzymes made of RNA, which catalyze RNA-RNA bonds, and thereby making a big step towards making the RNA world hypothesis more plausible. And then David skipped the usual postdoc and directly became fellow at the Whitehead Institute, and initially continued his research on origin of life and in vitro evolution methods, but soon became involved with projects which changed biomedical research fundamentally. So in collaboration with Tom Tuschl and Phil Seymour, he started characterizing and working out the molecular mechanisms of a new fascinating phenomenon, RNA interference. For this phenomenon, Craig Mello and Andrew Fire received the Nobel Prize in 1998 for observing that injection of a few molecules of double-stranded RNA is capable of robustly silencing cognate genes in C. elegans. And it was Tom, Phil, and David which found out that RNA interference is mediated by 21 to 23 nucleotide-long fragments, which form the basis from this double-stranded RNA, and this forms the basis of siRNA technology, which I'm sure everyone here used once in a while. And while characterizing these siRNAs, David's laboratory discovered, together with the labs of Tom Tuschl and Victor Ambrose, that animals and plants themselves produce endogenously an abundant class of small RNAs, of 21 to 23 nucleotides, which they termed microRNAs. And these microRNAs are potent post-transcriptional gene regulators involved in multiple physiological processes and diseases. And again, I'm sure that many of you have looked at microRNAs influencing your favorite process. And so since identifying microRNAs in 2001, David's laboratory produced a truly remarkable and constant stream of papers characterizing and working out the evolutionary biology of microRNAs as well as their molecular mechanisms, and forming the basis of everything, basically, which we know of microRNAs itself. So his lab was also instrumental in identification of other types of small RNAs, piRNAs, heterochromatic siRNAs, etc. So in 1996, David became an associate member of Whitehead Institute and an assistant professor of biology at MIT, where he is a full professor today. Of course, he was also a recipient of numerous honors and awards, among them the AAAS Newcomb Cleveland Prize, the Molecular Biology Award of the National Academy of Sciences, the Louis D. Prize of the Institut de France, and he was elected to the National Academy of Sciences in 2011. So today, David will talk about microRNAs and their regulatory effect. So please join me in welcoming Dr. David Bartel.
Thank you, Marcus. All right, so I really appreciate this opportunity to tell you what we're learning about microRNAs, and as many of you, I think, know, these microRNAs are these small RNAs that get processed from these hairpin precursors, okay? And from there, they get loaded into what we call a silencing complex that consists of the microRNA and an Argonaute protein. and then the microRNA directs the post-transcriptional downregulation of mRNAs and other transcripts. So we now know that, just looking at the mouse knockout studies where people have gone in and knocked out a particular microRNA or a family of pyrologous microRNAs, that often these microRNAs are conserved among many species, that these have defects in the development of skeleton, different microRNAs, some have defects in development of skeleton, brain, neurons, muscle, heart, lungs, vascular, pancreas, intestine, skin, fat, ovaries, testes, each of the hematopoietic lineages, as well as cellular and physiological behavioral effects. And this is what we had anticipated, because we know that most of the human mRNAs are under selective pressure to maintain their pairing to microRNAs. Most human mRNAs are conserved targets of microRNAs, and so it's going to be hard to find a disease or developmental process that isn't somehow influenced by microRNAs, and the mouse knockout phenotypes bear that out. And many of these developmental and physiological defects cause mortality. So there can be severe or other severe conditions like epilepsy, deafness, retinal degeneration, infertility, arthritis, immune disorders, and cancer. And some of these knockout strains also have altered susceptibility to infection or differential responses to models of disease or injuries. Okay, but as with many discoveries that are of fundamental importance to human health and disease, the very first microRNA was not found in humans or mammals. It was found in a model organism, and in this case, it was in C. elegans. So in 93, Victor Ambrose's lab was studying a gene that was important for the transition from larval stage 1 to larval stage 2. And he found that that gene did not code for a protein. Instead, it coded for this Lin4 RNA. And with genetic data from Gary Ruvkin's lab, it was clear that that RNA was repressing the output of this Lin14 messenger RNA. And that repression was needed to get from larval stage 1 to larval stage 2. So that was the first one of these small RNAs. And then the second was LET7. And this is found by Gary Ruvkin's lab, also important in going from making this developmental transition. In this case, a later stage in development. In this case, it was repressing the Lin41 mRNA. And LET7 is quickly recognized that this was not just in C. elegans, but there was also LET7 in flies and humans. Basically, all bilateran animals had a LET7 that also came up late in development. And there's this idea that maybe there are other small temporal RNAs, other RNAs that are important in the timing of development. And then, as Marcus mentioned, a year later, my lab, Victor Ambrose's lab, and Tom Tuchel's lab found that these Lin4 and LET7 were actually part of a much larger class of RNAs. They resembled Lin4 and LET7, but most were not expressed in a temporal manner. So it's beyond the timing of development. And because we had identified these molecularly, rather than through genetics, we really had no idea what mRNAs they might be targeting. But we knew that they were small. And so we called them MicroRNAs. And this is when a lot of people got excited about this class of small RNAs. At this time, we knew that there were over 100 microRNA genes in worms, in flies, in humans. We also found them in plants. And so essentially, any mRNA that a biologist might be interested in working on might be regulated by these microRNAs. Not just RNAs that are important in development. And so the, whoa, I did something wrong there. So the question then became, okay, what are these mRNAs? What are the targets? And so in plants, what we found was that the targets have very extensive complementarity to the microRNA. And so in plants, you have this extensive pairing, and that, in this context, we have extensive pairing. The microRNA directs the cleavage of the mRNA, kills the messenger, and of course, is a very effective way of downregulating the mRNA. In animals, you get some of this, okay, this cleaving of the mRNA. And this, of course, is reminiscent of the RNAi pathway, which Marcus mentioned, where instead of having RNAs coming from these defined hairpins, where you have essentially a single RNA specified by each of these genes, instead, in RNAi, you have this long, in endogenous RNAi, where you have a long duplex that gets chopped up, or hairpin that gets chopped up by dicer into these siRNA duplexes, each of them giving a small RNA that can get loaded into Argonaut. And here you, in the sort of the canonical, simplest form of RNAi, you have this slicing of the mRNA. So this, and this, of course, is very important. This is found really throughout eukaryotes. In many different eukaryotic branches, you have this RNAi pathway. MicroRNAs are thought to evolve from this pathway, and lots of interesting biology here going on, often to silence transposons or other heterochromatic regions, and also viruses, et cetera, a basal pathway. This is, in this sort of branch here, sort of maintains that really original basal function where AGO slices the mRNA. Now, in our cells, we have actually 4 Argonaut proteins. Argonaut 2 is the one that's able to do this slicing of the mRNA. The other 3 are only able to do this. But the fact that we've retained this slicing, of course, is very nice for those of us who want to do gene knockdown experiments. The RNAi pathway, this, you can find this, but it's pretty rare in vertebrates. It's seen in mouse oocytes, but not really in other somatic cells. So this RNAi can happen, but it's quite rare among in mammals. But all of our cells are using the microRNAs, and of course, because they can do this cleavage, that's the reason that synthetic siRNAs work in mammalian cells. You can synthesize these RNA duplexes, which we call siRNAs, but our cells are thinking of them as a microRNA duplex, and they go into the Argonaut, make the silencing complex, and then are able to cleave the target. Of course, they also have off-target effects, which are going through this pathway. So this other pathway, which is much more common, has much less pairing, and here we do not have slicing of the mRNA. So the Argonaut doesn't need to have this active site that slices the mRNA, and instead what happens is you have other types of post-transcriptional downregulation, which I'll get into later. The targeting here really depends primarily on pairing to what we call the seed of the microRNA, nucleotides 2 through 7, often supplemented here by a pair to nucleotide 8 or an A here across from nucleotide 1. So it's these short regions of pairing that specify the targeting here. Surprisingly little contribution from the 3' end of the microRNA. And this is the type of targeting that really dominates in mammalian cells, although I'll talk a little bit about this also later. All right, so this type of seed-based pairing, this canonical pairing, we actually found, everything I told you about it so far, we found computationally, okay, before we had experimental data for the targets of the mammalian microRNAs. And all we did is we just looked at the whole genome alignments at the regions. It's typically in the 3' UTRs where the microRNAs are acting. The regions within 3' UTRs that had conserved pairing to the microRNA, and then we looked at the types of pairing. We sort of imagined all different types of pairing that the microRNAs could potentially be using, and found that there was a particular configuration involving pairing here, perfect Watson-Crick pairing to the seed, that was conserved much more than you'd expect by chance. Okay? And so there are other types of pairing that were conserved, but only the same amount as you'd expect by chance. And so this is what really told us the importance of this seed pairing, and the importance, for instance, of this A here across from the first nucleotide, and these other features of targeting. And the reason that worked is because there are just so many messenger RNAs that are under pressure to maintain their pairing to microRNAs. Remember, most human mRNAs have these conserved sites to microRNAs, even when you subtract what you'd expect by chance. And so for each highly conserved family, and there are about 87 conserved families of microRNAs, each has on average more than 400 mRNAs that have a conserved site to the microRNA. Okay? Over 400 mRNAs under selective pressure to maintain their pairing to the microRNA. Okay? Now, often when an mRNA is targeted by one microRNA, it'll be targeted by another one. And sometimes these will be microRNAs that are expressed in the same cell type. Okay? And so when this happens, you get additional repression. As you add more and more sites, you get more and more repression. Okay? And on average, if you have a conserved site to a microRNA, every mRNA that has a conserved site to microRNA has about an average of 4 to 5 sites for the same or different microRNAs, usually different microRNAs. Okay? And so they can kind of add up, even though each of them is imparting a rather modest degree of repression, sometimes only 15%, 20% repression per site. But if you add up 4, 5, 10 of these sites, you can get quite large effects. In addition, there's a lot of non-conserved targeting. So later we had the data sets just looking at what happens when you perturb a microRNA, what happens to the proteins, what happens to the ribosomes, what happens to the messenger RNAs. And it's clear that there's a lot more non-conserved targeting than conserved targeting. Okay? So this adds up to over 60% of the mRNAs have conserved sites. When you include the non-conserved targeting, then even more higher fraction of the messenger RNAs are under influence of the microRNAs. And so if you're interested in the expression of a messenger RNA in humans, mammals, other animals, it's clear you're going to have to consider the microRNAs. At least at some point in development, they're probably going to be influenced by the microRNAs, so very widespread effects. And so to help people kind of place these into gene regulatory networks, we've been building computational models especially to rank the microRNAs, not only based on the extent of their conservation and how confident we are that they're a conserved target, so that's one mode that you can sort the targets, but also based on whether we think they're going to be more repressed than other targets. So we can rank them based on their predicted efficacy of targeting. And we've been increasingly improving these models of targeting to help those of you interested in looking at the microRNAs that might be regulating an mRNA that you've been working on. All right, so just one more kind of big picture slide here. The idea here is that the microRNAs are adding an additional layer of gene control. Okay, so a messenger RNA can be transcribed at different levels. Okay, the locus can be completely silent, or it can be very active, or somewhere in between. Okay, so you can have different levels of transcription, and then that mRNA goes into the cytoplasm, and it encounters the microRNAs. And the microRNAs, depending upon how many sites that mRNA has to the microRNAs that are co-expressed in that cell, you can have different shades of repression here, okay? And so in this way, you can get much more complex patterns of gene regulation starting with relatively simple promoters for the mRNAs and also for the microRNAs. So you can think of transcription as sort of setting up a column of gene expression. And the microRNAs are the artist, okay, the sculptor that's chipping away at that gene expression. Remember, they're always repressive, okay? They're taking out a little bit of that expression, chipping away at it, okay? And sometimes they chip away enough that you get this transition, this developmental transition, like going from larval stage 1 to larval stage 2, or other cases that have been seen in biology. So they can promote a transition, or they can decrease the amount of RNA that was there in a previous state and sharpen a transition. But more broadly, what they do is they produce a much more complex topology of gene expression, okay, to get a more optimal amounts of protein in each of the different cell types. And that tuning, if you will, or that sculpting away can, as I showed in the first slide, have, when you add up the effects on all the microRNAs targets, when you delete that microRNA, you can get very, very severe effects in the mice. Okay, so what I want to focus on today with respect to the newer science from my lab is what we're learning about this slicing mechanism, particularly in fish, and also what's going on in this mode. Okay, so starting with the slicing. Okay, as I was saying, this is the dominant mode of downregulation that you see in plants, but in humans, we've only found for all the microRNAs together, remember there's 87 families of conserved microRNAs, for all of those together, we found only about 18 targets that are downregulated through this slicing. Okay, so not very common, and you can see those here. Actually, the first one that we found was a target of microRNA 196. Okay, this is a microRNA that's expressed from the Hox cluster, and it actually targets this HOXB8 3' UTR through very extensive complementarity. Okay, just a GU wobble here, but other than that, very extensive Watson-Crick complementarity, and that's, you know, as you'd expect, it guides the slicing of the mRNA. Actually, and we found that evidence for that when we looked at messenger RNA or transcripts from mouse embryos, and we're able to detect in the clones that we looked at, 7 out of 8 of them map precisely where you'd expect if you had cleavage between the nucleotides that pair to nucleotide 10 and 11 of the guide RNA, exactly where you'd expect based on work from Tom Tuchel's lab, signature of slicing. So okay, that is what we found, but actually we initially had tried to see slicing in fish. It was easier for us to get fish embryos. We thought, okay, it should be there, and we never were able to see it, and now we think we understand why. So it turns out, and this is the work of Grace Chen, who's a graduate student in the lab, it turns out that fish, zebrafish, and actually all the other sequenced teleost fish, okay, and this is just 4 of them here, have 2 substitutions which dramatically decrease the ability to slice the mRNA. One of them is this glutamate going to an aspartate. You know, you'd think that would be pretty conservative, but it turns out to be quite important. And also this phenylalanine going to tyrosine, also quite conservative, but important. And so these 2 changes dramatically decrease the ability of the protein, the Argonaute protein, to slice the mRNA. And I'll just show you an assay here where Grace has injected a target of an endogenous microRNA into the one-cell embryo of zebrafish. And she'll inject that sometimes with a messenger RNA that encodes for more Argonaut. And remember, it's Argonaut 2 that's the one that has that slicing catalytic activity in human cells, mouse cells, et cetera. And so this is the assay. She injects the target, and if the Argonaut is loaded with Mir-430, this endogenous microRNA, you should be able to see slicing of the target. And we can just look at that on a northern blot. So here's her northern plot, and these are the markers where you'd expect the substrate and the product. And if she doesn't add any additional of mRNA, she sees that there's no evidence of slicing here. And if she adds more of the zebrafish Argonaut, she also does not see any slicing. But if she makes that aspartate to glutamate change or the tyrosine to phenylalanine change, changing it back to the ancestral sequence, now she starts to see slicing. She does both of them, and it looks like the human version of the Argonaut. And this is just quantitation of that. Okay, so we're able to get up to the human just by changing these two residues. There are many residues that have changed in the fish, but these are the two that have really reduced the amount of slicing. And these residues are it turns out to be very, very near to the active site. This glutamate here in the ancestral sequence is actually, we found this when we worked with Patel and looked at the yeast Argonaut structure and found that there was this fourth active site residue, this glutamate, that inserts itself into the active site in the active conformation of the protein. And then here's that phenylalanine. So just focusing in on this, there's this hydrogen bond network that sort of places this glutamate. And when you change this to an aspartate, of course, that's going to have an effect at the active site, or at least it could. And this tyrosine, we think, also somehow perturbs the active site. Okay, and so we can also look at this in a more sensitive assay where we just use the methods of Phil Zamore's lab to make purified Argonaut with a specific guide RNA, again Mir-430. And now with that purified silencing complex, we can just add in vitro with some magnesium and buffer, we can just add the substrate and just run out the products on a gel. And what we see is that when you have the human purified complex, we see slicing. When we have the zebrafish, we see slicing, but it just takes a lot longer. Okay, here we can see a fair amount of slicing, about 30% in 30 seconds, and here it takes 100 times longer to see less slicing. So there's about a 100-fold decrease in slicing in the zebrafish Argonaut. And that can be, again, it can be repaired by just restoring those ancestral residues. And if you take out another active site residue, it completely goes away. Okay, so this explains this absence of slicing, which is so important in RNAi. It explains why we're not able to see slicing of endogenous microRNA targets and why the technique of RNAi has been so lousy in zebrafish, okay? And many people had tried it when RNAi was first discovered and just were not able to get RNAi to work in zebrafish, and now we know that it's because the Argonaut is incapable of slicing the mRNA. And it explains why these morpholino reagents have been so popular in zebrafish, which of course have their own issues. The curious thing was, though, that what it doesn't explain is the processing of a kind of a unique microRNA called Mir-451. This is a microRNA that's important for erythropoietic development in the fish and in mice. And this is a microRNA that is too short. The hairpin is too short to be cleaved by dicer, and so instead the short hairpin goes into the Argonaut protein, and the Argonaut actually cleaves it in a reaction that's analogous to what happens when it slices an mRNA, okay? And then you have further resection and maturation of the microRNA. A very unusual pathway, but it requires this active site activity of AGO2, and it's known to happen in fish. Okay, one of these examples that discovered this was from Antonio Giraldez's lab, which was in the zebrafish. So Grace tested how well the zebrafish Argonaut could carry out this reaction. So in this assay, she just put in radio-labeled 451. In this case, all the samples had an mRNA that made a flag tag Argonaut. And so the experiment here is to inject this into the one-cell embryo, and then after 4 hours, pull down on the Argonaut, okay, so we can get rid of this, and then just look on a gel at, you know, how much is bound, and then how much is cleaved but not resected, and how much is kind of the mature RNA. Okay, so that's the assay, and here's the result. And what Grace found was that, okay, the zebrafish, although is clearly able to bind, okay, the substrate quite well, and it was able to process it. You can see that there's a fair amount of processing. It doesn't look 100 times worse than you know, the human here. Okay, part of that I think is because we're kind of outside the dynamic range of this assay, but part of it is just that somehow, for some reason, it's handling this substrate much easier than it is a slicing substrate. And as you'd expect, if you just restore it by changing it back to the ancestral residues, you can restore the maturation of this microRNA. Okay, so this has led us to take a closer look at this 451 sequence, and it's, you know, it's almost perfectly paired, but it has this GG sort of imperfection in this stem here, and this is the part that's going to become the mature microRNA. So at position 6, which is in the seed, a very important part of the microRNA, you have this GG mismatch. Interestingly, amniotes, so mammals, chickens, etc., they don't have this mismatch. Okay, and instead they have either a pair or a wobble at this position. But what we find, I don't have time to talk about to show you in detail, is that this is processed much more efficiently, particularly for the crippled Argonaut, than this. And this is a big surprise to us, and we want to follow up on this and figure out why it is that this is the first time that we've seen that a mismatch at the seed of the microRNA can actually enhance activity, but it really enhances the cleavage, not so much the binding, but the cleavage of this substrate. So we're following up on that, but that is the reason that the fish are able to make their red blood cells, make this microRNA, do proper erythropoiesis. Okay, so to summarize this slicing part of the talk, what we're seeing here is that it looks like effective slicing, effective RNAi, was lost in an ancestor of the teleost fish, okay, which is most of the fish that you see are teleost fish. In fact, most vertebrate species on the planet are teleost fish, okay, and yet they're able to do, this lineage is able to has done very well without slicing, without RNAi, okay. So if you're, you know, eating your salmon for dinner, you can say that fish did not have RNAi, and it's doing well. Well, maybe not that one, but in general, as a clade, these fish are doing very well without the slicing activity. And the reason that they've been able to get by with this very crippled slicing is because they maintain this GG mismatch within this microRNA. Whereas in the amniotes, we've lost that GG mismatch, but that's okay because we have a very efficient slicing Argonaut, and we can do okay without that GG mismatch. Of course, if you're a model organism, it's not that great not to have RNAi, and so and so now with the CRISPR technology and with this information, we think that we can restore RNAi to zebrafish now, fix the Argonaut, restore it to zebrafish, and maybe and just see what happens. You know, if restoring this activity that it hasn't seen for 300 million years. So, similar to experiments that we had done earlier with Saccharomyces cerevisiae, which had lost RNAi, and we were able to restore it. And of course, it'd be nice if it turns out to tolerate the restoration of RNAi. That could be then a useful organism, strain to be doing gene function, gene knockdown experiments. All right. So next, we've talked about the slicing pathway here. Next, I want to talk about this other type of downregulation. So when you have this seed-base pairing, or if the Argonaut just doesn't have the slicing activity, like AGO 1, 3, and 4 do not have the slicing activity in our cells, then you can still have downregulation, and it's through some other non-slicing type of mechanism. And the biochemists have worked out the dominant mechanism that's happening here, and that is that the Argonaut protein, which is brought to the 3' UTR by the seed pairing between the microRNA in these sites, recruits a scaffolding protein here. GW182 is the name implies, and that interacts with the poly A binding protein, and that recruits a deadenylase complex. CCR4 naught is the primary one here, which shortens the poly A tail, this deadenylation of the poly A tail. And what happens when the tail gets short enough is that eventually you have loss of the cap and 5' to 3' decay of the mRNA. Okay, so and our work has showed that decay of the mRNA is the dominant effect of the microRNA. Okay, there's also some translational repression. We can detect that too, but overall what we see is, you know, regardless of the cell type, whether the RNA is, the cells are stressed or translationally stressed, the cell type, the microRNA, et cetera. We've tried lots of different conditions, and pretty much everything that we see in mammalian cells shows us, and this is looking at ribosomes or proteomics and comparing the changes that you see at the ribosome level or the protein level to the changes you see in the mRNA. What we see is that the majority of the repression is explained through this mRNA destabilization. There's a little bit, you know, 10 to 30% in addition that's probably this translational repression, but the majority of it, regardless of microRNA cell type conditions, et cetera, is through this deadenylation, decapping, and decay of the mRNA. So actually, for those of you who've been in this field longer, you know that it used to be thought that microRNAs decrease the amount of protein without really changing the amount of mRNA. So that dominant translational repression just hasn't really panned out. And of course, this is actually good news for any of you who are interested in these microRNAs and want to know the targets of your microRNA or the effects of your microRNA, because you can perturb the microRNA and just measure the changes in mRNA and get a quite accurate readout of what the microRNA is doing. Okay, and that's a lot easier to do than looking at the protein or the ribosomes. It's much easier to look at changes in mRNA. So this is good news. But there is some translational repression. We thought, well, maybe the reason that these mRNAs would be translationally repressed is because their tails were getting shorter. And if mRNAs with shorter tails were translated less efficiently, that could explain, you know, really this whole deadenylation could be explained both the destabilization and the decrease in translation. And so we wondered, you know, to what extent does the tail length matter for translation? And actually that question hadn't really been answered. and that's because it's hard to measure tail lengths, okay, mRNA tail lengths. But in a few cases, particularly in early embryonic systems or in oocytes, it was known that there's a very strong relationship between tail length and translation. So this is just an example. The cyclin B1 messenger RNA, okay, during the maturing frog oocyte, the messenger RNA, the poly A tail, gets longer, okay? And when that happens, you get about a 100-fold increase in translation of that. And that's what's helped partly responsible for this maturation, okay? So this is at a time in the oocytes when there's no transcription, okay? So the way that the tail is getting longer is through a process called cytoplasmic polyadenylation. So normally our poly A tails, the poly A tails of messenger RNAs, are made in the nucleus, they go into the cytoplasm, the mRNA goes to the cytoplasm, and in the cytoplasm the tails just get shorter, okay? But in this context, you have a poly A polymerase in the cytoplasm that makes the tails of specific messenger RNAs get longer. And when that happens, you get dramatic increase in the translation of this mRNA. Okay. And it's thought that maybe this is more general, that longer tails equals more translation. And so we decided to look at that, but first we needed a way of measuring the tail length. So a former student of the lab, Alex Subtony, developed this way of using high-throughput sequencing to measure tails. And so what he did is he started with total RNA, ligated onto it an adapter using this split ligation here that appended this piece onto the end of the tail, and then sort of used partial nuclease digestion to, not complete, but just a nuclease that cuts after Gs to sort of trim away a bunch of the RNA, but we're mostly looking for RNAs here, expecting to have about 100 to 300 nucleotides of the messenger RNA remaining. And then he captures that based on this biotin, and then reverse transcribes, or adds another adapter and then reverse transcribes, and then puts the cDNA, without further amplification, just puts that cDNA onto an Illumina flow cell. So on the Illumina flow cell, you start making, for this high-throughput sequencing method, you start making clusters by bridge amplification on the flow cell. And so these clusters have about 1,000 strands each. So here's a cluster that's seeded by a cDNA with a very short tail. This one is seeded by a cDNA that had a somewhat longer tail. Then while we're still in the cluster generation station, we anneal a primer and then do a primer extension reaction where we add only TTP and biotinylated deoxy-UTP, okay? And that incorporates some biotins in here. Next, then, we take that flow cell and we put it on the sequencing machine and start sequencing. And that then gives us a read that corresponds to the very 3' end, sort of where the poly A was added, and the very 3' end of the messenger RNA. Okay, so now we know the identity of the messenger RNA, where the poly A site was, and then we wash and flow fluorescent stripped avidin and image again. And that reading, because we also put in standards and a standard curve, that reading of intensity, of that fluorescence intensity, then we can read out as a poly A tail length. So we get the mRNA, where the tail was added, and how long the tail was based on that fluorescence intensity. And we can do this for then millions of different messenger RNAs. And then we can map those messenger RNAs to transcripts and look at the distribution of tails. So this is just looking at 10 different genes, and you can see that here's one that has, and these are the tail lengths, and you can see that it has rather short tail lengths. Here, these get longer. Here, this one has much longer tail lengths. So this is just 10 examples. Of course, we have now thousands of genes for which the mRNAs pass are cutoffs. So we can look at this more broadly. And here we're just stacking up those thousands of mRNAs from thousands of genes based on the average tail length. Okay, that's the dotted dashed line here. So now you can see this distribution of distribution. Get a kind of a global look at the tail lengths for the mRNAs in this fibroblast cell line. And then we can look in different places. This is looking in yeast, either budding or fission yeast, plant leaves, Drosophila cells, mouse liver, okay, different cell lines. And then the most, I think, interesting examples are here in the embryonic samples. This is the fish embryo, and there's the equivalent stages of the frog embryo. And here you can see that in these embryonic samples, you have a broad range of average tail lengths. Okay, this dashed line really spans the distance here, but there's a lot of white space here and here. So for each gene, there's a rather narrow distribution of tails. Okay, and so this is what we kind of see overall poly A tails, which is nice to know. So then the question was, and we can have many questions, but the main question we had was, what's the relationship between the length of the tail and translation? We know that having a tail is important, but does it matter if the tail is 20, 40, 80, 160 nucleotides long? How does the length of the tail influence the degree of translation? And so for the translation measurements, Stephen Eichhorn got those, and so he did this by a technique called ribosome footprint profiling, and that gives us translation efficiency, relative translation efficiency on this axis, and then Alex's tail lengths on this axis, okay? And each of these points here are the values for the mRNAs from a particular gene, okay? So we're looking at thousands of genes here, and what you see in the zebrafish at 2 hours post-fertilization is exactly what you'd expect, that for these genes that have short tails, where their mRNAs have short tails, you see rather low translation, and as the tails get longer, you see much greater translation. Okay, you change the tail length. The twofold increase in tail length gives 5 to 10-fold increase in translation. Same thing at the blastula stage here, 4 hours post-fertilization, and then at gastrulation, it disappears. This relationship just goes away. Similar story in frog. Okay, you see a nice relationship here coupling between tail length and translation efficiency. At the earlier stages, at gastrulation, goes away. And then in all the post-embryonic samples that we looked at in these cell lines, no positive relationship between tail length and translation. In the liver, you can see that the Spearman R here is 0.00. This is not easy, okay? This is no relationship. Yeast, no positive relationship. Okay, so this idea that the length of the tail, the increase in the length of the tail increases the translation efficiency, turns out to be spot on in the early embryo, but not in these post-embryonic, not after gastrulation post-embryonic samples. Okay, so it looks like there's this developmental shift in the nature of translational control, where here it is coupled to tail length. And in work done, I've been showing you correlations here, but work that was done in the zebrafish, actually is in the frog oocyte, showed that this is causal. They could append a longer tail to mRNAs and see more translation. So we think that this is a causal relationship here. Longer tail means more translation, but then not so much here. Okay, and so there's this shift where the tail length is coupled, translation is coupled to tail length, and then that goes away, and this follows the shift in transcriptional control. Remember I said that early in these embryos there's no transcription, but then the zygotic transcription turns on, so now you have this maternal to zygotic transition in transcriptional control, and soon after that there's a shift in translational control, okay, which is coming around here. So what do we think is happening here? Why would you have these two different regimes, these two different regulatory regimes? Well, in this case, there's transcription, okay? You have plenty of transcription. That's a great way to regulate genes. Whereas here, all the mRNAs are inherited, okay? There's no active transcription off the DNA. And so, and in this case, there's also a lot of regulation going at the level of mRNA stability, okay? Some mRNAs are much more stable than others. Remember, that's what the microRNAs are doing. They're mediating these differences in messenger RNA stability, okay? Whereas in this situation, if there's no transcription, if you destabilize an mRNA, then the cell is not going to see that mRNA again until you have transcription. There's a lot less opportunity for differential mRNA stability. So in this regime, without these two mechanisms, and if you still want to have differential gene expression, what the cell does is it has this cytoplasmic polyadenylation, and then you have this readout of the tail length, where the tail length, oh dear, okay, good, where you have this coupling between the tail length and the translation. And this is pretty much, this was known already, okay? The surprise came here that this goes away, okay? That this coupling goes away. And the reason that we think it goes away is that if a lot of the regulation is happening through mRNA stability, that means there are going to be some mRNAs that have been in the cytoplasm a long time. And remember, when you're in the cytoplasm a long time, the tails just get shorter, okay, in the absence of this cytoplasmic polyadenylation. If the tails are just getting shorter, those mRNAs that have been in the cytoplasm a long time, well, the cell has kept them there for a reason. It wants to translate them. It would be a problem if the cell just started discriminating against mRNAs that have been in the cytoplasm a long time. Then there's no point in having differential mRNA stability as a mechanism for repression, for regulation. So if the cell wants to translate an mRNA that's been in the cytoplasm as well as it does an mRNA that just came out of the nucleus, then it's going to get rid of any coupling between tail length and translation. So the idea is that because this is so important in gene regulation, that you give up on using tail length as a way of regulating translation. Okay, so that work we had published here, we've now extended it to flies, and this is with Terry R. Weaver. The same story. Okay, now we're looking in changes as you go from stage 11 to stage 14 oocytes, nice coupling, stage 14 oocyte to activated egg, nice coupling, gastrulation, goes away. So this transition in the nature of translational control, it appears to be common to bilaterian animals, at least the few that we've looked at, and maybe ancestral to the bilaterian animals. Also, here's just looking even later, you can see there's plenty of change in tail lengths, but very little effect on TE, on translation. The nice thing about flies is we can look at mutants. So here's a case where we have the mutant in the cytoplasmic polyadenylase, the wispy mutant, and you can see that in this case, the tails, here's the wispy mutant, this is wild type, the tails all get shorter, but there's still a relationship between the tails that are longer in the wild type are still longer in the wispy mutant. And in fact, when we look at the translational efficiency, there isn't that much difference between the wispy mutant and the flies. So in the absence of so this is kind of different than what was seen in vertebrate embryos. It looks like in flies, the cytoplasmic polyadenylation is lengthening everybody, all the tails. But the differences between mRNAs isn't through this lengthening, it's probably through the shortening. And so those differences are maintained in this mutant, and the differences in translational efficiency are also maintained. Okay, so that was a bit of a surprise, something nice that we can see with flies. And I just want to close then coming back to the microRNAs. So remember, the reason that we are looking at this is that we wanted to know whether this shortening of the tail was the reason that microRNAs were less translated in mammalian cells. And we don't think that's the case because in the mammalian cells, post-embryonic cells, there is no relationship between tail length and translational efficiency. But what we're also able to do is understand a unique observation that had been observed in the zebrafish, where this is the exception to what I told you earlier. Remember earlier I said that you can read out the effects of the microRNA by just looking at what happens to the messenger RNA. You perturb the microRNA and just look at the messenger RNA, you can see what's going on. That is not true in this zebrafish embryo. It is true here and later. And originally this is interpreted as somehow reflecting the dynamics of microRNAs, but now that we know about this transition in translational control, we can understand what's going on. And we have experiments validating this. And the basic idea is that the microRNA comes on when you're at this stage, and at this stage, when the microRNA comes on, you get, of course, shortening of the poly A tail, and this is when the translation is coupled to tail length, and so you get translational repression. Okay? No change in the number of mRNAs, so now you would miss it. Just looking at the mRNA changes, you don't see any change. You have to look at the ribosomes to see what the microRNAs are doing here. At this stage, it's more typical of what you see elsewhere, where, the microRNA causes the tails to get shorter, and what that causes is for these shorter tail mRNAs, they go away, okay? And then there's a little bit of translational repression, but it's not because the tails are shorter, because there is no relationship between tail length and translational efficiency at this stage. Okay, so this explains the very different effects of microRNAs at these two different stages of microRNA. The two different stages of development. Okay, so we're quite excited about this result of this discovery of this developmental shift in translational control, and of course, we'd like to know how it's happening molecularly. You know, what is causing this what's causing this relationship early, and then why does it go away later? But for now, we can say that it certainly explains the different consequences of microRNAs at different stages in development. It's not due to the dynamics of microRNA regulation. It's not because microRNAs are doing something different in these different stages. It's just because they're shortening the tail, and that has different consequences at this stage compared to this stage. And this then explains why the early embryo is the only system where we've been able to see translation repression dominating as the microRNA effect. So then I just want to close by thanking the students who worked on the major projects I talked about. The loss of splicing in RNAi in fish, that was Grace Chen. Hazel is our zebrafish collaborator. And then Alex and Stephen did the poly tail lengths and translation, again with help from Grace and Hazel, and of course the fly work, and thank you for your attention.
In the past year, there was an article in Nature that referred to an article in Science about messenger RNA, and in the sperm, I believe, of mice that can actually transmit, that were found to be altered. The microRNA was found to be altered, and the sperm of mice who were stressed out, and then their offspring were also stressed out. And it was based on, now, there's only been one paper. I haven't seen anything in replication, so I don't know if this is a blurb or is something that is of value. Have you heard about that, and could you comment on it?
I haven't read the paper, so I guess I can't comment on it. Sorry.
Okay.
But it sounds like what they were proposing was that changes in microRNAs were somehow inherited.
What they were trying to propose was that the male can pass on stress in the microRNA of his sperm. As compared to controls. They did it with mice and controls. You could find I'm sorry, I don't know the author of the paper, but you could easily find it on PubMed looking up stress.
I can take a look at it. I think that it'd be interesting if microRNAs could be relayed from the sperm to the zygote. I don't think that is absolutely clear. There isn't a whole lot of microRNA repression happening until the MZT, until the microRNAs are actually transcribed off the zygote. And anyway, it's of course sounds interesting, but I can't really comment on it.
Thank you.
Yeah, sorry.
David, for your catalytically inactive agotoosin in zebrafish, did you look at the targets too? Does this give you a shift in the seed where you don't avoid 1011 anymore? Or does it have any benefit?
Yeah, it's a good question. We don't. There are very few mRNA targets that have that extensive complementarity, and so it's hard for us, because the numbers are so low. It's hard to us to make a statistical case for enrichment or depletion of those types of targets. But we really should look at.
Could you look experimentally now if in the fish actually you see more of these interactions, kind of as non-canonical?
Yeah, we could, but yeah, it's a good idea. We haven't looked at that. Yeah. But again, what we found more generally is that even in mammals, you can have a sort of a non-canonical site type that has about a 10 or 11 nucleotide match to the center of the microRNA, and that can work. But I didn't even mention it here because so few mRNAs have those sites. And maybe you're saying that, well, maybe more would have them in fish, but even in mammals, they don't mediate cleavage. It's not enough pairing to give cleavage. And so I don't think that the cleavage would influence the numbers of those sites. Maybe that's kind of the direction that you were thinking of. So I would be a little bit surprised if we saw a difference in fish, because even in mammals, don't mediate cleavage.
You mentioned that 70 to 90% of the repression is due to this destabilization of the mRNA transcript.
Yeah.
Well, what are the kinetics of this destabilization and the subsequent drop of the transcript, the abundance of the transcript? And maybe more specifically, if one was to do something like RNA-seq that's highly sensitive, how long after induction or introduction of an mRNA would one expect the levels of the transcript to drop to the point where you wouldn't detect them anymore, or you would detect a significant change?
We've looked at that for, we've made cell lines where we can induce the microRNA very, as rapidly as we can with an inducible promoter. We can also look at Mir-451, which is induced pretty rapidly. It's the most rapidly naturally induced microRNA that we've looked at. It's induced in hematopoietic cells when they're stimulated. So and then we can look at the targets. If they're after an hour, 2 hours, 4 hours, 8 hours, etc. So we've kind of looked at that, and the bottom line is that the longer the microRNA is there, the more repression you see. The mRNAs never go down to nothing. As I was saying, that if you just have a single site or just a few sites to the microRNA, you'll go down enough for us to detect, but not down to nothing. So you can see that, and we also looked at the same time using ribosome footprint profiling at the translational effects, and you can see some translational effects very early, but they're so small because the microRNA is so little at the beginning that we never really find a phase where the translational effects dominate, even when we look at the dynamics of the microRNA. So we don't really think that that's a context where the translational effects are going to dominate. Really, we think it's only in this early embryonic system when you have a different regulatory regime where tail length equals less translation, where the translational effects are going to dominate.
And the other type of experiments that you mentioned where you had fast induction, was it within 2 hours, 4 hours, or?
Yeah, we could see. It takes a while for the microRNA to come up, and within a couple hours after the microRNA comes up, we start to see a decrease. But again, to really see larger effects, we need to wait longer. So this is not the kind of thing that's going to give a very rapid response to some sort of extracellular stimulus or something like that. It's more of a, we think of it more as kind of sort of over the time course of development. You know, as a cell becomes a neuron or a muscle, it expresses these microRNA, these specific microRNAs that kind of create the milieu for gene regulation within that particular cell type.
Thank you.
Yeah.
Well, congratulations for the great MicroRNA review. I was told that the gene therapy might cost about $1 million per patient. So the lessons you described today, polyadenylation effect for increasing expression, translation of microRNA-induced effect versus the known suppression of the microRNA-induced effect, could be people using in gene therapy to avoid those areas that will repress the effect versus the polyadenylation?
I mean, there are efforts to inhibit microRNAs in therapy. There's a company called Regulus and other companies that do this. I used to be involved with Regulus, and their most promising candidate is actually a inhibition of Mir-451, very highly expressed in the liver, and that microRNA is important for replication of hepatitis C virus. And they can inhibit that in the human liver and cure hepatitis C with that, inhibiting that microRNA. So that's the thing that's furthest ahead in the clinic. But I don't know of anything where working on the poly-A-tail angle in the clinic. Thank you.
Perhaps I missed the point, maybe somewhere you said that, but for example, in very early embryos, the transcription, like in Drosophila embryos, zygotic transcription is not very active for a very long time, so until the stage 14. Yeah, so did you also say that microRNAs can be inherited through mother, or are they just start to transcribe very early that they need to?
Yeah, it's surprising how little microRNA regulation there is in the oocyte and zygotic transcribing embryos. After the zygotic transcription comes up, there's a lot of microRNA action, but before then, it's kind of interesting that there's a lot less. And so this tail length story that we're talking about, predominantly when we're talking about the oocytes and early ''embryos, in the oocytes at least, that's happening through other types of factors that are shortening tails, proteins that are binding 3' UTR causing tail shortening. And then upon zygotic transcription, you have a couple key microRNAs. In zebrafish, it's that Mir-451 microRNA that comes on, shortens tails, and that's really important, kind of, I guess at the stages I was talking about in zebrafish. But there are also proteins that are influencing not only the cytoplasmic polyadenylation, the extending of the tails, but also the shortening of the tails. Yeah.
In the study where you did the correlation between poly-A tails and translational efficiency,
yeah,
did you try to bin your mRNAs by size and see if they still don't have correlation? So, in other words, if the short and the long mRNAs have opposite influence from the poly-A tail, they could actually cancel each other, for example.
Yeah, so I'm pretty sure we tried that. We have a table in that paper with, we tried like 20 things, and I'm pretty sure that was one of the 20 things. But we could check afterwards. It's a good point, and we looked at things like half-life and lots of other things that might influence this relationship, and it's a supplemental table because we didn't find anything all that interesting, in that for those kinds of things. But we should look.
Been working on introducing small RNA-seq into some of our experiments, and in studying those library preparation techniques, it seems like a lot of them will make libraries for things that are greater than 23 nucleotides, up to say 200, but most of that gets wasted in order to focus on the 21 or 23 nucleotide transcript. So can you comment on your view of these non-miRNA small RNAs that are greater than 23 nucleotides?
We know from Susan and Gigi's work in bacteria that these are really important in that context as regulatory RNAs. In mammals, I think people have been looking in that space. They'll find a lot of small nucleolar RNAs there that might be doing interesting things, and other types of maybe unknown ones. So there could be other classes. A lot of times people are looking at just the ones that are more than 200, and we call those link RNAs or long non-coding RNAs, and then the small RNAs. But you're right, there is that intermediate RNA, and there could be interesting things there. Some people may be hesitant because there's a lot of tRNA there, and other things like that, but there might be ways of selectively, getting rid of that and looking in that area. Yeah.
Thank you.
So please thank David once again for this fantastic talk. See you at the